I made this one too. So we are going to start doing replication. And should I have to say DNA replication?
No.
Why?
Because replication means...
Because replication means making copies of the DNA. So, remember we said replication, you don't have to say DNA replication because that's redundant. The only thing that can be replicated is DNA. So replication is making copies of the DNA. When do we copy our DNA? Before mitosis. That's perfect. So I'm going to say before, first thing I'm going to say is, so before a cell divides. And when did you say that it happens? When is DNA copied? During the S phase. And what does S phase stand for? Yep, or synthesis phase. And that S phase is part of what? Interphase. Very good. And interphase is part of the whole... Yep, of the cell cycle. Very good. So we went through and we had to do mitosis and the cell cycle to understand why it was important for the synthesis to take place and when it happens. Why do we need to do it? Well, new cells will each need identical DNA strands. Remember, what are we making at the end of mitosis? Two identical daughter cells. Perfect. So those two identical daughter cells need to have identical genetic information. So just to take you back to like a minute ago. So we said that the synthesis phase or the S phase is during interphase of the cell cycle. Where does that take place? Where is the S phase going to take place if you're a eukaryotic cell? Well, let me ask you, what are we doing in the S phase? Nobody knows what we're doing in the S phase?
We're copying DNA.
Okay, making copies of the DNA. In a eukaryotic cell, where do you keep that DNA? What? In the nucleus. So it would take place in the nucleus of eukaryotes. I spelled nucleus wrong. What if you were prokaryotic? Like the nucleoid region. So if we go through here, G1, S, and G2, so the DNA replication takes place during the S phase, so DNA replication would happen there.
That's what that arrow is pointing to.
It's pointing to the S phase, right? What do we call the G1, the S, and the G2 combined?
Interphase.
So this is interphase. I forgot to label one of them. What is this last one? That's the M phase. Right? And M phase includes both mitosis and cytokinesis. What's the difference between mitosis and cytokinesis? Remember, mitosis is defined as nuclear division. What is cytokinesis? That's whole cell division. So mitosis and cytokinesis, they're similar in the fact that they're both divisions, but mitosis is just division of the nucleus, whereas cytokinesis is the division of the cells. So when we start talking about replication, we have to find a place to start, and the place to start is called an origin of replication. So the origin of replication, that's the starting place for replication to occur. And bacteria, how many chromosomes do bacteria have? I'm sorry?
20.
Okay. How many chromosomes do bacteria have? What does the DNA look like? They have a single circular DNA. So because it's single and because it's circular, bacteria only have one origin of replication. Now your DNA is in, what is that thing called again? What's the structure of your DNA? It's in a double helix. It's really, really long. So eukaryotes will end up having multiple origins. Remember, bacteria have the single circular DNA. Their chromosome is single circular DNA. So let's take a look at an animation of prokaryotic replication. We're going to have to do it in a different app because it's not going to work on here. All right, so DNA replication, where did we say it began? We just talked about it a second ago. You got to see it in the prokaryote. What do we call that place where it starts? The origin of replication. It's not a real fancy name. The names kind of tell you what they do. So it's going to begin at that origins of replication. And eukaryotic replication, remember we said we had more than one origin as compared to prokaryotes, which only had one. So we have two strands open up, and it's going to form, just like in that animation you just watched, it's going to form something called a replication fork. And this replication fork, it just looks like a Y, so it's Y-shaped. It's a great way for you to look at a picture and figure it out. And your new strands of DNA will grow at the fork. So if you look here, this is our replication fork. The whole thing looks like a Y. Yes, the whole replication fork. It kind of looks like a zipper opening. Because it's similar to a zipper. If you've ever watched a zipper on your coat go up and down, you see how like the little teeth fit together and it goes down, it can make a Y. It looks very similar to a zipper. If you pay attention to the numbering here on it, notice that one strand is 5' to 3' and the other strand is 3' to 5'. That anti-parallel nature, which allows those bases to fit on the inside. Right, think about when you watch that documentary in Rosalind Franklin. Remember how Watson and Crick thought, oh, we'll put the bases on the outside. And that made no sense. And then she looked at it and she told them, you guys are crazy. But because of the anti-parallel nature, and we know that purines always pair with a pyrimidine, we're able to get that nice compact double helical structure of the DNA. So as DNA replication begins, you heard in that, they talked about something called the bubble. So at DNA replication, you have, as the two strands start to open up at the origin, replication bubbles form. Prokaryotes, which are bacteria, you know, they have only a single bubble. Why would they have only a single bubble? They have one origin. Oh my gosh, that was so smart. Because they only have a single circular DNA. We'll see. Because they have one origin. Eukaryotic chromosomes, they have multiple origins of replication. So that means they're going to have many bubbles. So if you look, these are all these different origins that they're opening up even more. They're going to pull that DNA apart, and we would still have one going 5' to 3', and this one would be 3' to 5'. It's still the same strands. What we've done is we've untwisted it. We've gotten it into that ladder form when we looked at our DNA model here. We've taken at our origin of replication, we've started to break it open, and now we've got these bubbles forming that are going to stay there and allow for space for replication to happen. Yeah, it'll close up behind it. Yeah, that's why you see it forms a bubble, because after the zipper thing opens it, it kind of starts closing behind it. Remember, DNA is double-stranded. It doesn't want to be separated. So let's take a look at a simple animation for it. So the first enzyme we have to talk about is the one that unwinds and separates the two DNA strands. And its name is DNA helicase, or just simply helicase. Helicase. It ends in what? Ase, so you know it's an enzyme. What is it going to do? It's going to unwind the helix. Helicase. So it unwinds that helix, and it separates the two strands of DNA. And it's going to do that. It's going to have to break the bonds that are holding them together. What are those bonds that are holding them together? Are those strong, or are those? So by breaking the weak hydrogen bonds between the nitrogen bases. And just in case I've forgotten, we know a purine always goes with a pyrimidine. So I'm going to have A always goes with T, and G always goes with C. Who told us that? Chargaff. So it's also called Chargaff's rule, or complementary base pairing. Those two terms are synonymous. How many bonds are between adenine and thymine? Two. How many between guanine and cytosine? Perfect. The next enzyme that they had there is single-stranded binding protein. It doesn't end in ACE, but you know it's a protein and it's doing a job. It's an enzyme. So we have single-stranded binding proteins. What's the normal form of DNA? It's a double helix. So you've got two strands that are attached to each other, and DNA is most stable when it's at that double helix, when they're attached to each other. So what we have is single-stranded binding proteins, and these are going to attach to help keep those two DNA strands separated and untwisted. They're going to want to go right back together. So the single-stranded binding proteins are there to keep them pulled apart. So they keep the strands separated and untwisted. You like to think of the single-stranded binding proteins, it would be like if your best friend broke up with their significant other, right? And the single-stranded binding proteins, you know when they always get that crazy idea, it's like, oh, we should just get back together. Your job as the best friend is to be the single-stranded binding proteins and ensure that they remain separated, okay? So you are the single-stranded binding proteins for them. So another enzyme that we have to talk about is topoisomerase. I know it sounds crazy. Where's my friends who always ask, does spelling count? Guess what? It counts. Topoisomerase. Isomerase. So this is going to attach to the forks of the bubble. So it's going to relieve the stress on the DNA as it separates. To the relieve stress on the DNA molecule as it separates. So really, the topoisomerase, this is kind of like, oh, your best friend just broke up. You're doing your job as a single-stranded binding protein. You're keeping them separated no matter how hard they want to go back. But it's really, really stressful, and they're really upset. So then topoisomerase would be like if you took them to like the spa for a day, and they could surround themselves with big comfy pillows and towels, and it helps relieve the stress of the situation.
But that's kind of awkward taking both of them to the spa at the same time. Oh.
You can take all your friends if you'd like.
No, I'm
So if I took Anthony to the spa.
no. Not for example.
So much.
I don't like spa.
It's like if I took Spencer to the spa. That's weird. I don't want to take
There's nothing wrong with gentlemen going to the spa. You can care for yourself. That's right. So before the new DNA strands can form, you have to have a place for them to start. So in order for wait a minute. How are we going to copy DNA? DNA, what are we attaching to the inside of it? New nucleotides, right? So before we can do that, we need to put down a primer. We got to get it ready. Kind of like when you're going to paint a wall in your house, and let's say that for some unknown reason, you painted your bedroom hot pink before. If you wanted to paint over it, you would have to paint over it with a primer to get it ready. So the primer that we lay down is, well, it's got to be a nucleotide who doesn't mind being single-stranded. What kind of nucleotides don't mind being single-stranded? Do you know a nucleic acid that's single-stranded and not double-stranded? Well, there was DNA, and that one likes to be double-stranded. RNA. So we have to lay down these RNA primers. These RNA primers are going to give us a place to start the addition of new nucleotides. And of course, if we're going to lay down RNA primers, we need to have an enzyme that does it. So remember, if it's an enzyme, generally it ends in ase, and the name tells us what it does. So this one here is named primase. Primase means it lays down the primers. It's not first because we had helicase unwinding it. We had those single-stranded binding proteins keeping them apart. We had the topoisomerase relieving the stress. Helicase, single-stranded binding proteins, topoisomerase. Yeah, that's what I just said. And then primase comes along, and it's going to lay down our RNA primers for a point of attachment of our new nucleotides. And then finally, primase is going to go ahead, it's going to synthesize or make the RNA primers, which are just RNA nucleotides. Finally, we're going to then add the new nucleotides. Our new nucleotides are going to be out of DNA. So we need an enzyme that's going to do that. We're forming a big DNA strand. So we're taking many nucleotides and putting them together. So what do we call that molecule where we take monomers and we make a whole bunch of them together? Polymers. So and wait, what type of polymer are we making? We're making RNA polymers? No. DNA. So the name of our enzyme is actually DNA polymerase. Yep. So DNA polymerase, it makes a polymer of DNA. How do you make a polymer of something? You add a bunch of monomers. What's the monomer of DNA? Nucleotides. So DNA polymerase tells you exactly what it does. So if you take a look at this really easy to understand picture, yoink.
Oh, I thought you were being serious.
So if you take a look, we've got our helicase going down this way. So it's unwinding and separating our double helix. You've got your origin of replication up here. That's where replication is going to start. On the outside, you would find your topoisomerase and your single-stranded binding proteins doing their job. We've got primase coming here, putting down a primer because DNA polymerase doesn't know where to start. It has no place to attach to. So we've got to give it a place to attach. So notice down this one strand here, we've got only one primase. In this strand, we've got 1, 2 primases. That's going to end up being our lagging strand, the one that we synthesize or we make discontinuously, or meaning not in a row. Why do we have to use the primase anyway? Wait a minute, who's adding our nucleotides? Right, so DNA polymerase. The reason we need those primers, DNA polymerase can only add nucleotides to the 3' end of the DNA. What that means is, if you remember before, I'm really bad at drawing.
I would have given up by the circles.
So what molecule is this supposed to be? Rudimentary. It's a nucleotide. Here's my phosphate. Here's my nitrogen base. Is this one purine or pyrimidine? What? A pyrimidine. How did you know? Just a single ring. Very good. And then this guy here, this is my 5-carbon sugar. In this case, if we're talking about DNA, it's my deoxyribose. Remember how we number those carbons. If we go starting at the oxygen, we start labeling it in a clockwise fashion. So this first carbon here is going to be my 1'. This one's going to be number 2. This one's going to be number 3. This one's going to be number 4, and this one's going to be number 5. So when we talk about saying it can only go from the 5, it can only attach to a 3' end, we're saying that it can only attach down here. So because DNA can only add nucleotides to the 3' end, this means that the new strand can only be built in the 5' to 3' direction. So if you look here, this is my 5' end, this is my 3' end. Your DNA is you have your RNA primer here. Who laid down my RNA primer? Primase did, thank you. Then DNA polymerase is coming and adding on my new nucleotides, but it can only add it to the 3' end of my template strand. So the one that they're building off of is called a template strand. So I'm going to go ahead and label this as a template. What does a template mean?
It's like a guide.
A guide. That's a great word for it. It's a guide. It's telling us where we to start and where we put it down. How do they know which nucleotide to put here? Right, so they have the base here. So if this base here is A, what's this one going to be down here that we're putting on? A T. Very good.
But for RNA, doesn't it have uracil?
It does have uracil instead. You're absolutely right.
Like switch.
It's going to, uracil is like having thymine there, so that's going to attach to the adenine. So what we like to say is that we say that DNA is synthesized. What do we say synthesized means? Made. So DNA is synthesized 5' to 3'. So here's our new strand. It's going to be 5' going down to the 3' end on a 3' to 5' template. And that's based on the numbers of the carbons. So how is DNA synthesized? 5' to 3' on a 3' to 5' template. That is honestly the only thing my husband knows about biology. He has heard me say it so many times that is the only thing he knows. He's like, ask me how DNA is synthesized. That's the only thing he remembers. He has no idea what it means. He just knows the words DNA is synthesized 5' to 3' on a 3' to 5' template.
Why is that the main thing you say at home?
It's not. He's just heard me say it to various students over the years. So if it's so important that my husband knows what it is, it's probably really important that you guys not only know it, but understand what it means. Again, going back to how we labeled these guys. Remember the 1, the 2, the 3, the 4, the 5. The DNA can only add it down here on that end. So what does that look like in a molecule? Well, remember, the strands are anti-parallel, which means that one goes 5' to 3' and the other goes 3' to 5'. So if this were my strands of DNA, which one would we be able to lay it down on? Which way would it be the new strand being added?
It would be added on
You would have a new 5' one going right here on this side. So this would be my leading strand. And then I would lay another one down here, and then I would lay it down here, and that's the order in which it would go. So when we make our new DNA strand, the leading strand is synthesized or made as a single strand from the point of origin, because that's where we got started, remember? Towards the replication fork. So again, remember, DNA is made 5' to 3' on a 3' to 5' template. So it's going to be made in this direction. This is my leading strand. How many strands in DNA go 3' to 5'? Only one, because it's anti-parallel. So we've got to do something special to the other side.